Bom dia a todos. Então vamos iniciar agora a nossa sessão. E primeiramente eu gostaria de convidar o professor Dr. Terence Dwyer. Ele é epidemiologista, professor da Universidade de Oxford, e na Austrália também ele trabalha no Murdoch Research Institute. E eu vou convidá-lo para falar sobre o Consórcio Internacional do Câncer da Criança, um esforço mundial. Doctor.
Thank you very much. Thank you, Sylvia, for your introduction too. Your meeting today is about environmental causes of children's cancer, and you will cover many aspects of that, I know, from legal aspects to social and political aspects. I am a scientist. Scientists provide the knowledge which is the basis of any legal or social political action. And in the area we are in, which is childhood cancer, the science is not nearly finished. There is much for us to find out. And my talk today is about one major global effort to understand more about the causes of childhood cancer. So this is a study about how to prevent childhood cancer. Science can also provide information on how to treat it, and in fact we have done very well in learning how to treat childhood cancer. As I will show you, we have not yet done very well in knowing how to prevent it, and this study is one way of getting more knowledge, and I believe it is critical to us understanding how to prevent childhood cancer. Oh, before I go, these are the people involved. You can see on the left a large number of names up there who are involved in steering this work. You can see Sylvia Brandelise is there. Oh, okay. And also you can see that Campinas is one of a number of countries that are involved in this work. Now, I don't think I have to convince this audience that childhood cancer is important to Brazil and to the world. Should I stand back a bit? Okay. These are the major causes of death in children. And you can see that cancer is one of those major causes. But this doesn't tell us nearly how many children get cancer. About 5 times as many children get cancer as actually die from it. But it is still a major cause of death in children. But despite how important it is, and I draw your attention to the bottom line here, we only understand 10% of the causes of childhood cancer. And this is despite the fact that, for example, this work here was done 50 years ago. The National Cancer Institute in the United States, which has the most funding of any institution in the world for this work, has made this statement that in the last 50 years we have made very little progress in understanding causes of childhood cancer. Now, part of the reason is that this is a complex puzzle. I've put up how I think about the work that I do. I am an epidemiologist and I focus on collecting data on humans who get disease. But I try to relate it to pieces in the puzzle that come from other research, cellular research, genetic research, research on animals. In terms of childhood cancer, we are going to need to know a lot more about humans than we know now if we are going to solve this puzzle. Sometimes the answer can come from just genetic studies. For childhood cancer, we are going to need human data. And how would we get that human data? Well, the way people like me, epidemiologists, Closer? Yes. Oh, I thought it was echoing. The way people like me get data on humans... Sorry. Is to study how disease occurs. And of course, disease starts at some point. For children's cancer, it will start when they are very tiny or even during pregnancy. Something will cause the cancer to start, and then they will develop it as children. And I've got a diagram here showing that these are the children who in concept develop cancer. That's what we're interested in. But we actually have to either study those children and compare them to children who don't get cancer, which is one approach, or we have to start with a large number of healthy children, study all of them and wait till some get cancer. So far, the approach of looking at children who have cancer and looking back in their lives to try to find out what might have caused that cancer and comparing them to the children who are still healthy at their age, that has been the approach that has been used. If we had looked forward, we would have called it a prospective cohort study, hence the name of our consortium, the International Childhood Cancer Cohort Consortium. So that means we are doing studies that start here and wait till children get cancer. But all the studies to date have been these ones that look back. This is a very common approach that medical researchers use, and it's proved to be very valuable. And in childhood cancer, it has to be used. It's an important approach. But when we look at medical research in general, we do tend to do these we include prospective studies, which in our case, with childhood cancer, would start here, go all the way through to here. In adult cancer and adult heart disease, these prospective studies have been part of the story for 50 years, and they have provided very important answers and ways of understanding what is going on that the studies looking back can't always do. If we did an adult study of 100,000, it sounds a lot. You will see it's not enough to study childhood cancer. We would find that in that 100,000 adults, every year, 100, depending on where you were and what the disease was, which type of cancer or heart disease, 100 to 700 would get the disease. So after 10 years, you would have somewhere between 1,000 and 7,000 people who developed the disease and on whom you had information. Childhood diseases are much rarer. Some, like sudden infant death syndrome, that I was involved in running a cohort study on almost 30 years ago, was more common. Now it is much less common because we were able to find a cause, which is that putting babies on their stomach caused about 80% of the deaths, and those cases do not occur so much anymore. But for childhood cancer, we would need at least 10 times as many individuals, babies, studied as we would adults. And 10 times as many means 10 times as costly. It's hard to get money to do medical research on adults. To get something that costs 10 times as much is something that is prohibitive. I hope that's translatable. And in fact, when I was thinking about doing this study, when I was at the National Institutes of Health in Washington, D.C., back in 2004, 15 years ago, I did some calculations and worked out that if we truly wanted to get enough cases to study childhood leukemia, we would need about 1 million mothers and babies on whom we collected information. That should stop people in a way, and it has until now. This is why there have been no prospective cohort studies on childhood cancer, because you need to measure too many babies. So one way to approach this is to say, well, let's just do the retrospective case control studies where we might measure 1,000 mothers and babies and get the information we want. Why study 1 million? Well, part of the problem is for some of the things that we want to investigate, And I know this group will be interested, for example, in pesticides. It proves very difficult to measure it accurately when you ask the mother questions about her past or her husband's past. Were you exposed to pesticides in the years before your baby was born? This is a German study that shows that if you ask the mother those questions and then try to estimate the risk associated with pesticides for childhood leukemia, you find if you ask the question within 4 years of the child's birth, you estimate that the pesticides increase risk 4 times. If you ask it 6 or more years later, you say it only increased risk by half, by 50%. These are big differences, and they suggest that maybe even this is not accurate. In other words, when the mother recalls, she can't remember accurately, and maybe she recalls, if she has a child who developed leukemia, she even exaggerates a little in her mind, or maybe the mother who doesn't have a child who has leukemia doesn't think hard enough and underestimates her exposure. It doesn't matter what the effect is, we can't rely on this data for pesticides. Doesn't mean we can't rely on it for everything. This is another area that I have been working on in Oxford, which is maternal. This is maternal and infant infection. There are lots of reasons for thinking viruses in particular might lead to cancer. But when we try to collect data from humans, we find this happens. And this is British data from a study of around, this is a retrospective study of around 10,000 infants. And one of the things they did is they looked at social contact outside the home. What were the social networks for the child, play groups, that sort of thing. They found if they asked the mother whether the child had such exposure, she said they had a lot. So these social exposures increased the risk of infection. And if you took the mother's report, you would have estimated that those social exposures reduced the risk of childhood leukemia. About 40% reduction, that 0.6. If there was no effect, it would be 1. If it's 0.6, it means reduction. But when they use the GP records, and in England all the children and families have general practitioners, and they do surprisingly record information on things like social contact for the child in early life, the GP records gave exactly the opposite answer. They said that having the social contacts didn't protect the child, they increased the risk. And when they went directly to history of infection, they didn't find quite the strong associations, but the direction was exactly the same. The mother's report, if you had relied on it, would have said infection in early life protects, but the GP record said no, infection increases risk. So using the retrospective data for these 2 exposures, and I'm not saying all exposures, but for these 2, is going to prove to be too unreliable to rely on. Would that be better? You think? Okay. Can you hear me? Could you hear anything I said before? Okay. So, in 2004, I was asked by the National Cancer Institute to give advice on whether the study of 100,000 babies in the United States could give an answer to childhood cancer. And my answer was no, it is too small, 100,000. But there are other studies in the world, other birth cohort studies in the world collecting data, big studies as big as the one you are planning in America, and if we got them all to join forces, while none of them could solve the questions of childhood cancer, if they all participated and pooled their data until we got 1 million mothers and babies, it could be done. And they were convinced it was worth doing, and we started. And we now have 6 participating cohorts and 12 that we call emerging, meaning that they have expressed interest in participating, but we don't yet have data from them. Some are starting to give us data, but the 6 here have all provided data, and that includes the large studies in Norway and Denmark of over 100,000, smaller studies in England, ALSPAC, which for those who work in the paediatric area is quite famous, and the study that I was involved with that looked at sudden infant death syndrome in Australia. So these are the studies we have, and an older US study and a big one in Jerusalem. And here are the emerging studies. You will see that Campinas is listed there. I need to find out much more detail on this visit. Sylvia passes on what's happening, but I know you have made a lot of progress in the last year or two, and I'm very interested in that. There are other studies in other places. The Northern Italian study has just started to transmit data. This study in China, in Guangzhou, have told us they will transfer data next year. And as you'll see, we need them all to transfer the data. Where we are up to at the Where we are up to at the moment is that this number has now passed 700. We have 700 cancers that have occurred in almost 400,000 babies that have been enrolled and for whom data is available. About 200 leukemias. And in this group, the participating cohorts, by 2020, we think we'll have this number. In the emerging cohorts, these are the numbers we would have by 2020. And you can see that if they all participated, we would have the million we need. The total numbers of cases would not be all we needed because many of these they're very early cohorts and the cancers have not all occurred. We have to wait another 5 or more years for that. And these are the measurements we have. Go down here. For those who are interested in environmental chemicals and other factors, occupation is a very important way to find out about that, and all our cohorts have that information. Pesticides and chemical exposures, the big cohorts in Denmark and Norway have that information, and some others, but we will have to rely also on the new cohorts to be able to investigate that problem. But as you can see, there is a lot of information from these cohorts, and we are very fortunate they have collected so much so well. Now I'm going to come towards the end of my talk by showing you just a few results we have, because you'd think after 15 years we would have solved this, but this is not a 15-year endeavour. This is more like a 30-year endeavour, and people like me have to live long enough to see it out or pass it on to others. The first hypothesis we tested was this one: is birth weight related to childhood cancer? And I've just got some photos here of twins, just to show that even twins can vary a lot in size. Babies do vary a lot in size. And the reason we studied this is not because it was novel. We studied it because other studies, including the case control studies, the ones looking back, had studied it and shown us an association. Bigger babies have a higher risk of all childhood cancers, including leukemia, which is the most common. So this is what we found when we did our study. We found that in all our 6 cohorts, just marginally here for Denmark, the bigger babies, what this shows is that this would be no association, this would be a protection, and this, if it's above that line, it shows an increase in risk associated with birth weight. When we pooled it, we found there was a significant association, and the estimate was that for every kilo increase in birth weight, risk for all cancers was increased by 26%. So a meaningful result. Other people had shown that. What we don't know, what no one knows, is why that is. There are theories, and I could give you the theories, but in the time I have, I won't. But what could it be that's associated with birth weight that might be causing the cancer? Well, we thought maybe about nutrition, and we thought, well, in the cohort studies, we have information that is reliable that we can look at that might give us a clue. And in science, and in my type of science, you're always looking for clues to put into that puzzle and go to the next stage. So up here we have two things we looked at: the size of the mother, the pre-pregnancy BMI, and the weight gain during pregnancy. Two different things. What we found using our data was that pre-pregnancy BMI was not related to risk of cancer in general or leukemia. But if we looked at leukemia and acute lymphoblastic leukemia for weight gain during pregnancy, we found there was a significant association. So what we are now doing is other studies to find out what that means. What is it about weight gain that might be important? I'm not going to say to you that we are absolutely certain this is correct. There's much more work to do, but this is the sort of information we're able to get out of our data set. A second hypothesis we have looked at is birth order. Simple to measure and can be measured in the retrospective studies. And what we showed is not what everyone has shown. For birth order, the findings are not as consistent as for birth weight, but in our pooled data, this is the firstborn risk, which we set at 1, and this is the risk for secondborns, thirdborns, fourthborn, fifthborn. And you can see this is birth weight and this is birth order. This is kilo of weight, a kilo baby, weight baby, 2 kilo, 3 kilo, 4, 5 kilo. This is firstborn, second, third, fourth, fifth. And you can see that for birth order, it's like birth weight. Risk decreases with birth order. No, this was birth order, sorry, my mistake. This is birth order adjusted. This is for all cancers. This is for acute lymphoblastic leukemia. My apologies. But what it shows is that birth order is associated with reduced risk, and we don't know why. We think maybe it's because of the risk of infection, varying risk. A second and third-born baby is more likely to be exposed to infection because it has an older sibling who will be exposed to infection. Oh, sorry, wrong way. Now, the other thing we found is that when we looked at the data and we looked at birth weight and birth order together, first of all we looked at birth order, and this is just a summary. It's not first versus second, third and fourth. It's just the firstborn versus those who are second, third and fourth all together. And we found a reduction in risk of about, that says 22% reduction. But when we put birth weight into our statistical model, we found the two interacted in a way that's demonstrated by this. For a lighter baby who was later born, risk was reduced much more. We don't know why this is, but a light baby who was second, third or fourth in order had about a 70% reduction in risk. We are now investigating why this might be so. Another clue. Now on to things that you are particularly interested in, and this is very early, our colleagues at the National Cancer Institute in America have been analysing the data, and they've been looking at, via occupational exposure, not direct measurements, occupational exposure, looking at pesticides. Why fathers rather than mothers? Because in our data set, the mothers don't report a very high exposure to pesticides. They might be wrong. It's not because of their recall, because the information is collected well before the baby gets cancer, but they might give wrong information. But there aren't enough exposed to pesticides in our data set. For fathers, there are more. Even there, it's not a very high proportion. So the number of cases we are looking at that we can use to estimate risk is low. And the only significant finding we have is for acute myeloid leukemia. And it looks important, but it's based on so few cases that we need all these other cohorts to start providing data so that we can show that this is true or not. But we are working on it. Now, to finish, where are we going now? Well, one of the things about prospective cohort studies that is so important is you can collect biological material before the disease occurs. And we have done that using cord blood. We've taken blood from the baby when they're born, from the umbilical cord, and it's been stored. These are our Japanese collaborators, where they have their biospecimen storage facility, which is very sophisticated. And what are we going to do with the biospecimens? Well, this is the concept. The concept is that, and this word, the exposome, was developed in the World Health Organization Cancer Agency in Lyon, the International Agency for Research on Cancer. And this is a combination of questions and basic measures with analysis of the biological material. And what we're hoping to do is better measure chemical exposures, to get information on mechanisms that might underlie exposure to something, and even to find molecules that might predict cancer risk. Have we done anything? Well, we've started. This year we will be looking at the metabolome, and people may not know much about this, but if you take blood or urine, you can measure all sorts of molecules these days that couldn't have been measured 20 years ago. We can measure things that relate to food intake. We can measure chemicals, PCBs and dioxins. We can measure hormones in the blood. There's so much we can measure now on humans that we couldn't measure when I started my work as a medical researcher 40 years ago. So we're going to be doing this in the next year. We're also doing a project now involving researchers at Oxford where we're not looking at human DNA and genetics, we're looking at the DNA of viruses in cord blood to see whether viruses in cord blood can be measured and through their DNA, and whether this is related to childhood leukemia. And we're also going to look at what's called epigenetics to see whether we can find a pathway. Epigenetics helps us find pathways. So, second last slide. I'm talking to you as a scientist. Why would cancer specialists, children's oncologists, be interested in the sort of thing that I am talking to you about today? You know, we can treat effectively many children. They come through hospital and they're treated, and thank goodness for that. When I was a medical student, about 90% of children who developed cancer died. Today, 80% are cured. Isn't that enough? Well, those who work in hospitals would know that if you could avoid the suffering of the child during treatment and their families, you would. You prefer them not to have to go through it, whether it's successful or not. So, one, if we could prevent it, that would be even better. The other thing is, the children who are treated, there is evidence that there is a higher incidence of other diseases later on than for a child who hasn't had cancer and gone through treatment, and we would prefer to avoid that. The other thing I find, and I worked in a paediatric hospital before I went to Oxford, parents ask questions. They want to know. They say, is this something I did? You know, did I do something that caused my child's cancer? Could I have avoided it for them? So they want answers, which we can't give them now. And there's a possibility of screening children for high risk of cancer. Maybe Kel is going to talk about this, I'm not sure. Kel Schmigelo, who's here. But we can now identify different types of childhood leukemia, for example, that are due to genetic abnormalities. What are called chromosomal translocations, where the genes are carried, the chromosomes split and reattach in a way that's harmful. And it's possible, this is work that's going on at the moment, it's possible that these abnormalities, probable that they arise during pregnancy, maybe in 1 in 100 fetuses, and maybe more than 1 in 1,000 of these might develop leukemia. This might be a means of screening very early in life for those children who later might develop leukemia, but there is no value or little value in screening if you can't do anything. It just raises fear about the future. So we need to know how to do something if we screen these children. And to finish, that's why we need this work to go on. We will work on the 400,000 babies that we already have data on. That will continue, and we'll do things that I suggested, like metabolomics and looking at viruses. But we'll only do it on 700 cancers and 200 leukemias. We need the new cohorts to start providing data so we have enough information to find answers that will help us prevent childhood cancer. And that is something I'm devoting my efforts to with the help of people like Dr. Brandelise. Thank you very much for listening to me.